Hey all, welcome to Share Trek. This is Raj here. Friends, I met up again with Dr. Eric Arts, who was kind enough to give some time for us from his busy schedule. As you know, he's been working on HLP, and I wanted to talk to him about some exciting new approaches that have been undertaken for gentle myeloablation or clearing out the bone marrow for stem cell replacement. And I was trying to speculate if that technique could be extended to do bone marrow transplant for HIV patients. So I wanted some expert opinion. Dr. Arts agreed to join us, and the interview that I have follows. So at the end of this video, I'll be back again to just summarize my thoughts. Let's get started. Yeah, thank you so much, Dr. Arts, for giving me some time. And our audience also is always looking forward to information from experts like yourself. And the topic I had today was basically outside of the HLP, because I know you're working very hard on HLP. I know the research funding situation and all that. So the movement, I suppose, is going to be slow because of the funding activity for HLP, I guess.
Yeah, I mean it's actually progressing relatively well right now. But I would have to say from the US side, it's going to be a challenge
Right.
given the change in administration and lots
Yes.
of crazy stuff. But I'm more hopeful in the last couple of months than I have been. So yeah.
Right. So what I'm thinking is that we should probably meet again for the HLP also, because now that you mentioned there is progress going on, we could schedule one for a later time. And as we were discussing about this new technology that is being tested out in preclinical, I was hoping that we would ease into that. And to begin with, I wanted to just talk to you about just a rehash of what the HIV community was looking for when those critically ill patients, like the Berlin patient and the London patient, they got bone marrow transplant because of blood cancer, and that being the ultimate treatment available. And they recovered, and we discovered CCR5 mutation. Everybody in the community got excited. And later on, they got a bit frustrated because even now, I get questions in the comment, Raj, what's going to be the cost for a bone marrow transplant? And can I get that? So I would like you to explain to our audience from your perspective why it is not recommended, and so that we can set the constraint as the backdrop on which we can discuss the new technology.
Sure. I think, it's unfortunately for most HIV infected individuals, people living with HIV and on long-term treatment, of course they would like to see a cure. And in many people, they are willing to take a lot of even risk, I would say, in achieving that cure, so that they can live HIV free. But there is what is, I would say, an acceptable risk in terms of health, and then ones that you probably are putting yourself too much at risk. There's a reason why gene therapy in general, or anything that has to require like a bone marrow transplant, a complete myeloablation to remove your immune system and redevelop your immune system with a donor, it's all with considerable risk. And it's not something we take lightly, or anything that I would advise. So it's different when you can do gene therapies where that isn't required, where you can target something very specifically with gene therapies when you know you have, for example, a marker on the cell surface that says, okay, that's maybe where a latent HIV virus might reside in that cell. And then you can focus on targeting that cell. And then you're not requiring to get rid of all of your other immune cells to prevent it from coming back in some ways. So that's it's kind of a very layman's description of it. And when we get into aspects of gene therapy or those cell targets, it gets very complicated because they're coming at you from all disciplines in biomedical research, right? So sometimes you're finding something that you can target that people in cancer are more familiar with because they're things that are markers that appear on cells that normally don't appear on cells. And you can say, okay, well, we can get rid of those cells because those markers shouldn't be there. And those same markers are there because HIV is infecting that cell and latent. Then you see that again is something where you can target, but it's not always that is even an acceptable risk. So it's complicated.
Because I was reading up on the CRISPR Therapeutics website because they did the sickle cell disease therapy,
Yeah.
and we were covering it in our investment channel. So I was explaining to my audience how the patient experience is. So I believe there is almost a month of hospitalization first to release the HSC cells and then collect them, make a backup group and a group for actual modification, genetic modification. And then they are put through myeloablation, and they are under strict control in the hospital to make sure they get blood and that they are not infected. And then all these gene editing is done on the cells, stem cells, and reinfused back. It's autologous, reinfused back into the body. And the whole process requires around 3 months to 4 months of hospitalization, and it's very disruptive. But because of sickle cell disease having those VOEs happening twice a week or twice a fortnight, it makes it worthwhile to go into that risk. Whereas for HIV, ART is giving you almost close to normal life. So most practitioners would not recommend a bone marrow transplant, and CCR5 mutation is so rare that you can't be guaranteed that when you're ready that there's going to be a donor with CCR5. So that said, most HIV patients are still looking forward to that miracle cure, which is going to give them functional cure. Of course, we got HLP going on, we got EBT 101 going on, AGT 103. All of those are going on as research and clinical trials. So having looked at that risk reward aspect, I just wanted to share a document with you out here so that our audience can also see what we are talking about. And that happens to be this one. So I hope you are able to see my screen.
Yeah, it's coming up. Yes. Yeah.
Yeah. So one of the challenges that I find as a layperson, as a content creator for genomics, is that most of these companies do not give full information about, and of course they would not, because layperson doesn't have the technical knowledge and skills to understand it. But even the sketchy information is not there. But what I gather is that this particular therapy, Exacel, and for SCD and Exacel for beta thalassemia, already approved by FDA, and their patients are already being treated. So that's already settled. Now, what they're trying to do is they have got two candidates, one in preclinical trials and one in early stage. And the first candidate is basically trying to target HSC cells in the bone marrow and perform the edit so that sickle cell variant can be suppressed. The second one seems to be targeting CD117 and delivering a cytokine directly on the stem cell. This is what I gathered by looking at documents elsewhere from reputed sources like NIH and other areas. But I can't definitively say that what I read there matches what it is shown out here because there is not much description. But assuming that this were the way I am describing it, then I wanted to check with you if you see any possibility of a therapy for HIV, the way that they are doing for sickle cell disease, where you're targeting CD117, which I believe mostly appears in stem cells and some cells like mast cells and others. But as long as you restore the stem cells, any of the subsequent cells that are destroyed can be regenerated. So, do you see any theoretical possibility at all, speculating that you could have a therapy like what CRISPR is attempting for sickle cell disease, but instead to modify the stem cells so that the CD4 T cells created from there may have a suppressed CCR5 receptor?
Yeah, so you know, so from a perspective of you know, aspects related to myeloablation or just basically removing stem cells so that you can repopulate with a donor stem cell. Um, like those technologies and those therapeutics are getting better and better all the time. So most of the time now, this is not a process that is associated with a risk of mortality, even as it once was. And so you see a lot of people going through that myeloablation and repopulation and even targeted removal of cells, which is good. And for the most part, that's really important for cancer when you can do that and various forms of cancer, but also especially immune-based cell cancers like B-cell lymphomas, et cetera. And sickle cell as well. Like I work at a in Uganda where I was when you interviewed me last
Yes,
time.
last time,
Uh,
yeah.
they're developing a gene therapy institute there to treat sickle cell disease. And that's one of the big frustrations is that, sickle cell disease is not really that frequent in high income countries. And so there has to be an approach to bring this to low resource settings. And when you start implementing it in low resource settings, you find that there are ways of improving costs and making it really somewhat cost effective. And like you say, with bouts of sickle cell coming frequently, it makes a whole lot of sense to
Yes.
treat those individuals with this type of approach. So with CD117, c-Kit, it's the same kind of thing. It's a really effective way for removing the stem cells and then repopulating. The thing is that, you're still repopulating with a foreign donor. And it's not an autologous repopulation. It can be, but most of the time it's not. And as a consequence, there's so much risk associated with that in terms of long-term consequences. Most of the time, you don't suffer from graft versus host disease, but aspects related to that can still come into play, which can negatively affect your health. And the other thing is when you bring in CRISPR technologies where you're replacing a specific gene product and then transfusing. Um, we still don't know the long-term risks of that. So as much as we screen, and I would say we, the royal we, I don't do any of this, right? But as much as you can, there's always the possibility that one cell sneaks through. And we've this has been observed in lentiviral-based gene therapies. For glioblastomas, for example, it makes sense because the prognosis in that case may be 6 months, and extending an individual's life in with a gene therapy is is reasonable.
Right.
But when you use it earlier on, there is developments of certain cancers that are unexpected, and that's partly because of the lentiviral sort of integration, maybe near an oncogene in a very few, very infrequent number of cells. So when you look at CRISPR, I'm always concerned of off-target effects. And I think for diseases that are really detrimental and impact the livelihood of individuals and the quality of life, it makes total sense.
Right.
But when you can maintain a high quality of life without that approach, and you can live lifelong, like with HIV infected or the people living with HIV now, that even long long-acting antiretrovirals where the burden of dosing and pill burdens are all kind of eliminated. There's minimal adverse events and side effects now
Right.
that, you're the one thing that is true, and this is the reason why I still work in the area, is because you stop taking your drugs and you just resume your disease course. So that's where the frustration, or not the frustration, that's where my concern lies. And
Right.
unless you have more and more data on long-term outcomes, then you have to be very wary about approaching this when you could die sooner than you would if you were receiving the antiretroviral drugs alone. So,
Right. Yeah, I think from when you look at it from that perspective, it definitely makes sense how we have to exercise a whole lot of caution, and probably there'll be a whole lot of clinical trials to be done. All the side effects and everything have to be seen, both for the in vivo as well as the autologous approach, assuming we have the capability to modify the donors, the patient's own stem cells, and suppress the CCR5, which is a long shot, I suppose. But assuming all those things are possible, still it'll only be worthwhile uh, if the condition was such as you described. The quality of life is suffering, life expectancy is suffering, unless you do the bone marrow transplant. Those things are going to come to a head. In those situations, it makes sense. But for HIV, I mean, you couldn't say it is fortunate and unfortunate, because people have ART, which gives them good quality of life. That is fortunate. But unfortunately, because of that being the case, recourse to things like bone marrow transplant, myeloablation, all those things become very high risk considering their current status quo. That's the way I'm understanding what you have said. And
yeah,
the second fact you mentioned is very important, is cost factor. Many people in Africa don't have the kind of money that is needed for these fancy procedures. And I suppose that's where the HLP approach appeals a lot to me, because what you are saying is not only should it be inexpensive to make, it should be easy to transport. You need not have to refrigerate it, and you should be able to administer it easily without having a special setup. So when you contrast that approach to what we are discussing right now, this seems like something really with bells and whistles and everything, where you just need a bicycle. We are talking about a bulldozer.
yeah. Yeah, I mean, but just to, expand, from HLP, there is new. I mean, we just recently hosted Nicolas Chaumont, who is from University of Montreal, and he is a world expert in HIV cure. And so he just had a paper, I think, last year on metformin, and it's targeting of an mTOR pathway that, really potentially could assist in a cure approach. And it's a very targeted circumstance, but there were challenges with that as well. But those are also, there are more and more that we're learning every day, that there are changes in cell physiology when you establish a latent HIV pool that we're just coming to grasp with. And if you identify key markers that identify those cells, then you have a better way of figuring out how to eliminate them. And that could be a very targeted kind of gene therapy, a activated dendritic cell approach. All of these types of possibilities exist, but it's different. You're not requiring a complete removal of your immune cell population in order to go after what remains.
Right.
And that is the key, I think. So, I working a little bit with Vive on these inhibitors of apoptosis, SMAC inhibitors. Um, these are types of approaches that might have benefit as
Right.
well, and we want to use it and a lot of this is going to be combination-based therapies in the end as well, just like you have with HIV.
Yes.
And I mean, with antiretrovirals, but the idea is to just have it for a short period of time, and then, see if a patient or a person or a patient in this case would be can go on remission. So I think it's really hard because you get a lot of contact from people living with HIV on long-term antiretrovirals, and they ask me when can they enroll in a study, for example, with HLP, and I say, well, it in some ways it looks promising. It's just unfortunately these developments just take for so long, right? And there's
Right.
so many things so many hurdles, right? For good
Yeah, you have
reason.
to also think through it carefully because you're using valuable resources and time and the lifespan of your career within which you want to make sure that you optimize everything. So you want to dot all the I's and cross all the T's to make sure your strategy is right, the methodology is right, you have selected the right patients, you have the right time for ATI or whatever you may need to do. All that involves a lot of thinking. And before you commit anything, you want to be absolutely sure that 3 years down the line, you're not going to regret a decision that you took earlier, I guess. I mean, that's the way I see it from a layperson's point of view.
Yeah, no, I agree. Like you could have the most effective drug, the most effective candidate, and if you use it in the wrong way, so dosing, for example,
Yes.
and it is the case with any cure-based strategy, and it's so much more the case than treating for HIV because you are going after the needle in the haystack. And if you don't set that right,
Yeah.
and I think a lot of times, a lot of approaches that we've already used therapeutically have not been optimized. And they threw it at an individual and said, okay, is this going to work? And unfortunately, it none of them have.
Right.
But I don't know. Some of them, I also look at like TLR7 ligands, and I'm thinking, well, maybe if they were approached a little different and delivered in a targeted strategy, they might have worked better. But
Right.
you know, you kind of you kind of dissuaded any development of those anymore because of the way they were initially approached. So
Yeah.
yeah,
Yeah, I think you have a point there because we interview Jeff Galvin on a regular basis from American Gene Technology. And yeah, he explained to us how they did their phase 1 clinical trial. So what they are doing is they are harvesting the CD4 T cells from the HIV patient, and then they are doing some modification internally to suppress CCR5, and they incorporated a siRNA inside to disrupt any replication, and they put it back into the patient's body. So they didn't do a ATI for at least 2 years or something. And then they did an ATI. So the good thing was that they still found that their modified CD4 T cells had persisted, though in a smaller number, but they started multiplying. But the
yeah,
viral load started picking up immediately. So they had to stop, and now they are trying to do phase 1B or phase 2, but they don't have the funding at this point of
yeah,
time. They are raising the funding, but they have a better plan of how to do the ATI because now they have the experience. 15 days after the dosage, immediately do a ATI and let the CD4s, modified CD4s multiply, replicate, and all that stuff. But again, now funding is a constraint in this current environment. So to the point that you were making, if you don't have a proper approach, a good promising candidate can also probably fizzle out and maybe nobody else picks it up. So yeah, I hear you on that one.
yeah. It's a. Yeah, I just wrote a commentary for it. What looked like it was going to be a very promising inhibitor that could have been used in cure strategy too. It was in molecular therapeutics, and it was on a CD4. It was an immunoglobulin where the FAB domains of the immunoglobulin were taken off and put CD4 on in a modified form that was really working effectively in targeting HIV and looked like a very promising drug. and it still is in some ways because you could see how it could be delivered. You're not going to get necessarily a host response that would eliminate it. But it's just like everything in therapeutics, you have the problem that resistance somehow always emerges. And and that was the case here. And so you again, I think the key is to get a few of these together and to work effectively in a combination, and you should be able to achieve eradication.
Yeah,
But it's like the old days in HIV is that every company feels that they can develop a drug on their own or a mechanism on their own and as a consequence, none of these are doing enough. That's the problem.
right.
And so, I mean, when you looked at the early days of AZT, because the desperation of having an antiretroviral drug, it kept AZT in the course of treatment for decades after that, which wasn't necessarily a great drug, but because other drugs came and added to it, it was in combination therapy for the longest time, right?
Yeah. And also, I'm seeing these strategies which are targeting only the conserved areas to avoid this drug resistant thing. But monotherapies have frequently been consigned to being drug resistant over a period of time, small period of time.
Yeah.
So combination is good. One of the reasons why I was very excited about these two approaches that we saw on CRISPR therapeutics, extrapolating them from sickle cell disease and transplanting it for HIV, was the fact that we have these macrophages which might be infected with HIV, and they reside in the brain. So the blood-brain barrier is effectively, to a great extent, preventing ART from having an impact in the brain. And I was thinking that if we have a liquid nanoparticle that could have an anti-CD117, and if that could have some editing payload like excision, which cuts HIV in two parts and makes it into three pieces. If something like that was there, maybe that could pierce the blood-brain barrier and potentially give us the chance of a permanent cure with the kick and kill approach also being put together. As you said, one approach may not work. You have to have multiple things going together. So that's why I was very excited. But I see all the practical problems that you're talking about, like the HSC, by the time it changes and becomes a CD4, there are so many stages that it goes through. How do you do the edits, and how do you prevent the ripple effects from happening if a cell becomes something else? So I can see all of those things. So I wouldn't say that I'm disappointed, but I would say that it means that for most HIV patients, they have to mentally prepare themselves that the research community is attacking it on the life stages, it's attacking HIV on the conserved regions, and it's attacking HIV with various kinds of approaches, gene therapy approach, small molecule, large molecules, everything. So sooner rather than later, something good will come out of this. And I think the lowest hanging fruit now seems to be once in 6 months ART probably is the next target that might come to be true. And probably that is as good as it gets until approaches like yours and AGT and Excision manage to get through phase one, phase two, and come closer to being available to people in the arms. What do you think
Yeah.
of that?
Yeah, what you suggested, a targeted sort of gene therapy, something where you would deliver a payload that is antiviral, but not antiviral in the sense that it inhibits replicating HIV, but goes after it. Even if you want, so in those circumstances, if you're willing to sacrifice the cell that has latent HIV in it, or even some cells that don't have latent HIV, then you can survive that circumstance, right? And there you're targeting a CD4, CCR5 population. And if you find some other markers, then you say, okay, we're going to go in and we're going to change the nature of that cell to a certain extent and hopefully it's not going to be that detrimental and it's not going to kill all those cells, but it is going to chop up the genome that is in those cells, the HIV genome. And as a consequence, it doesn't pose a threat anymore. Um, those types of things are feasible. That's the type of thing. And there are some really good studies that are coming out. Uh, Matthias Lichterfeld's studies in at Harvard right now, where he's really diving into all sort of markers in the latent pool, everything that that cell might express. And it's complicated because what it expresses is combinations of markers that all cells express in some ways, but there are clues because there'll be multiple markers that identify that as a cell that might have latent HIV, and it's usually long-lived memory T cells, which is not unexpected. But
Right.
if you say, okay, if you're looking at a long-lived T cell that is, let's say, HIV specific, and you find a way to get into it, or not even HIV specific, just a long-lived T cell that is specific to some antigen and you target it, then it may not pose a problem for your longevity, right?
Right.
There might be a way to do it, and then you get rid of the HIV in that aspect somehow. And so those approaches, I think you'll see more and more. I kind of hope that we move away from the massive, you know, myeloablation,
Yeah.
stem cell transplants, because
Yeah.
it's like what we talk about, repeatedly, is like
Yes.
this is okay. So the biggest concern I have right now is PEPFAR. If PEPFAR
Right.
does not
Yes.
maintain their funding status,
Yes.
the 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 consequence for the world is beyond the understanding of most people. I just lectured on this. I said, they were responsible for half of the people around the world receiving treatment. So even if they take a break and they don't provide treatment for 6 months for individuals that have been on long-term treatment, we already see what the levels of drug resistance to dolutegravir is.
Yes.
If that starts emerging, and even let's say a quarter of those patients develop resistance, even a tenth of those patients develop resistance to dolutegravir, all bets are
Scary.
off because then you have millions of people that can transmit again, and we won't be talking about cure anymore. We'll just talk about, can we get the next antiretroviral drug developed?
Yeah,
So
I think I realized that I was going to talk to you about that. I mean, I was thinking how we could structure these questions without getting political about it, because right now the environment is like anti-expertise environment where experts in a field are not being consulted, and it's assumed that they don't know what they are doing. And I think we are seeing that happening. If it's localized to a particular country or a particular government, that's a different issue. But when it comes to HIV, as you said, all these drugs, they took a long time to go through the preclinical, clinical approval stage and then into the arms of people. And a lot of work has gone to make people aware of it and get them onto those drugs. And thanks to all those things, the spread of HIV has reduced. And if any of these drugs HIV becomes resistant to any of these drugs, then all that investment is gone. And
yeah,
do we have new medicines coming up at the rate at which we have developed? I don't think so. So everybody is in risk. Everybody, before you go to your dentist or before you go to operation procedure, you would start asking whether you sterilize this or that. I mean, as a layperson, I would be afraid. I don't want to have something infecting me because I've gone in for a surgery, despite whatever might be the case of standard protocols for sterilizing equipment or whatever. So we are looking at a very difficult future if these cuts continue longer? Because right now, I think that in some places there will be a little bit of stockpile of medicines that can go. I think they have got some stock for until February end or something. But as you rightly pointed out, if funding stops, that's another big challenge. We'll be running to stay
Well,
on the same spot.
but we don't, it doesn't have to be a political discussion because I was just writing an op-ed just the other day just and if you look at what high-income countries have provided to the Global Fund, and you compare that to what the U.S. has provided to PEPFAR, and Global Fund and PEPFAR are kind of half and half in providing treatment globally. It it the rich countries in the world are not contributing their share compared to what the U.S. is contributing. It's
Right.
very often, and I think you know. We have to do our own part. We can't just look around and say, well, the US is giving up half the funding for this, so we'll just meet it. And yeah, it might require doubling or even tripling the budget to meet what the US provides, but that's not that much when we look at our own GNPs, and it's understandable. So I mean, so you run the risk of if I send out this op-ed, it's going to look like I'm, you know,
It's
like
the
if
fact.
PEPFAR ends, it will be devastating for the world, and even for 6 months, it'll be horrific,
Yeah, it'll
and
be horrific. It'll be
and people
horrific.
don't
Yeah.
realize that, right?
Yeah, I mean it's the same thing that we face with environment also, right? Environmental scientists are crying out from the rooftops about global warming, and we are not doing anything about it. We are always looking at short-term gains. It's like we are going on a long journey in a car, but we don't want to stop, fill up the radiator, do preventive maintenance, and then move ahead. We just want to
Yeah.
do one trip, and we are going to reach the end. So yeah, I share your anguish, and I think many of the people who are watching our videos, they also share. One of the reasons why, Dr. Arts, I wanted to have this discussion with you is because as far as our audience is concerned, they are like they wanted this cure as of yesterday. And when we have this discussion and explain to them why certain avenues are not as popular for coming up with a functional cure, it makes them manage their expectations much better, so that they don't start hankering after this bone marrow transplant and all these fancy things, because at the end of the day it costs money. And to carry forward the point that you mentioned, most of these countries can also come up with their own policy approaches to buffer this shortage of funding by doing some, for example, communicating to people about prevention and other things. They could use it in the postcard or something. There are things that they could do without extra costs, using their own mechanism, like a thing running on the bottom of the TV in all programming, which talks about prevention of HIV or something like that. And maybe other countries like European Union, Japan, and others can contribute money and make the shortfall to come up. So those things are also possible, but allowing this to create drug-resistant strain is very irresponsible.
Well, yeah, and my biggest fear is, we have a population of close to 27, 28 million that are on treatment and have viral suppression and that are available to receive a cure. And I see it for the individual, but I also see it from a eradication of the pandemic perspective too,
Right.
right? So, and I think when I see that opportunity being lost because of, let's say, not continuing antiretroviral treatment globally, then it's heartbreaking, right? It's you just kind of like, I've spent 30 years working in Africa. I don't want to see this happen.
Right.
Um, but but that's my own selfishness, I suppose, or my own, I mean,
No, I don't
global.
think it is. I don't think it is selfishness. I have a analogous example. It may not be, it may be a bit crude, but basically, when somebody sees a piece of meat in the supermarket, right? That is one level of distancing from the reality that a live animal was actually killed. And, and then when one sees it in a restaurant, in a plate, already cooked and neatly arranged. So there is
Yeah.
this distance between reality and the presentation. When you go to Africa and actually see the human suffering and families who are haunted by HIV, and then you come back to the developed world, you can see that they are poles apart. I think many people in the United States and Canada probably don't realize how terrible this disease is and how dangerous it could be if it came out of the enclosure, which is basically becoming drug resistant. If they knew, I don't think they would play around with this. I think I heard somewhere that Marco Rubio was saying that AIDS, art medicine would still be funded. I'm not sure if it was finalized because right now I don't know what is happening, but hopefully they reinstate at least ART funding so that diagnosed people can continue to get the medicine.
Yeah, I think there's instability, but I believe that I have to believe that you know things haven't gone so awry that just general understanding and decency for the world is
Yeah.
still there, right? And so I think we would be facing so many other global sort of catastrophes if they decided to
Yeah.
cut this because it says that they don't care or nobody cares anymore. I don't think that's the case. So
Yeah,
anyway,
let's hope that better angels prevail and everything goes well. So, Dr. Arts, from your point of view, given the current environment that we are in, and your personal experience with the challenges of going through a successful research, setting it up and getting it done, and your experience in Africa, you have a global kind of outlook. What are your thoughts that you would like to share with our audience with regard to the landscape of HIV research and HIV therapy? Just a broad question. Any thoughts that you have on mind that you want people to understand?
yeah, so I think, I think people on um, on treatment now and that are adherent and that have sustained suppression of viral load, I think they could see a potential cure in their life. And I don't necessarily say that that's going to happen in 5 years. It probably definitely won't happen in 5 years, right? You might have a therapeutic that looks promising, but most of us realize that it takes close to 10 years. If it doesn't exist today, it might be 5 to 10 years before it actually appears in the future or longer. But I do think it's on the horizon. And I think even I sometimes with CRISPR and gene editing, I might sound less optimistic, but it's not less optimistic. I just think we still have further to go. And I think once we get over certain hurdles, we have a lot of possibilities in the near future. Same thing with CAR T cells and other cell-based therapeutics. I think it's the future of therapeutics. I don't think chemical-based drugs are going to be common in treatment in the next century. But from that perspective, I think it's hard. It's really hard for someone to be patient and wait as these things look like they're promising, and you're just kind of saying, well, why isn't it going faster? And the other thing is when you see this incredible media that is surrounding people that are actually cured. Well,
Yeah.
there's about a dozen people now that potentially have either a cure or a remission, but the possibilities could have been in 1 to 2 million in high income countries that could have had that same advantage. But those are the few individuals, all of the right conditions to obtain that cure. And usually it was because they were very sick. And they would have died if they hadn't received that treatment,
Yeah.
independent of HIV. So they really shouldn't look at that as a possibility for themselves, but the possibility that a cure is possible and that it will be obtained. It's just going to take some time. And that's the best I can.
Thank you so much. Thank you so much, Dr. Arts. So that I think our viewers, on behalf of our viewers and myself, I'd like to thank you for taking time to talk to us among your busy schedule. And hopefully we can get together again when your time permits to talk about HLP, when you have some information that you would like to share with us. And we'll do that. So thanks a lot once again. And we'll see you again when we meet next for the HLP, I suppose.
Well, it's always a pleasure and fun to talk with you, Raj. So thank you.
Well, friends, I hope you enjoyed the discussion that I had with Dr. Eric Arts. And as always, he's been kind enough to share his thoughts and give us a guidance on how to look at HIV therapy research, the way you need to look at it in order to manage your expectations. He articulated all the challenges that would be on the way and how they plan to overcome it. He also spoke about his concerns about PEPFAR funding and possibility of drug resistance if funding was stopped, even for a short period of time. So those are all valid concerns. And at the same time, he mentioned that it's not all doom and gloom. We have to be realistic, and a cure is coming for sure. But expecting something to come out of the bone marrow transplant the way I was thinking about it is a bit premature at this point of time. And so that's the gist of what we had as a discussion. And of course, we're going to meet with Dr. Eric Arts again later on to discuss about the progress with his HLP research, which he mentioned that fortunately he has been able to cobble together the financials that is needed, and it's not too bad from his perspective, as Dr. Arts said. So hopefully, maybe in a short while, we'll get in touch with Dr. Arts again and have a discussion around HLP. Till then, bye for now.